that's where all those terms come from. So we'll go through those and then remind me again at the end to go back through where all those letters come from. Okay, so when we're talking about direct versus indirect, so there's a couple different reasons why we use direct versus indirect. So we have two different ways of testing our patients to see if our patients have something. We have one way that you'll have thousands of micro wells of these trays with the wells in them, and say for example you are working in a lab, you're a clinical lab scientist, you're working in a lab, and you have thousands for HIV. Well, you've had 7 patients coming in to get HIV tests this week. Okay, cool, I can get one of my wells out and I can test immediately if all those 7 patients all at the same time if they're HIV positive. Or, I don't know what my patient has. My patient's got some sort of virus. What am I gonna do about it? How am I gonna test? Am I gonna test all the viruses? No, I'll probably try and find a faster and easier way to be able to tell what my patient has. So there's a couple different reasons why we use direct and indirect, and I'll walk you through those. So I have a cute little cartoon I'll show you too, but direct, what we're looking for is when you add the patient's sample to an empty well. So that well, it's sticky on the bottom, but there's nothing in it. So what you're looking for in direct ELISA is you're looking for an antigen. Does your patient have the proteins or something in there from the pathogen itself? So that's what we're looking for in direct ELISA. We're looking for the pathogen. So what would happen is, for example, you don't know what the patient has, and you just put the patient's sample in a sticky well. First, they would take the patient's sample, they would take their blood or urine or whatever it is, sputum, and then if it's blood, for example, they'll centrifuge it so that they take out the red blood cells so they only have the plasma on top, and then they can use that plasma to see what is in there, what is in the blood. So then we put the patient sample in the wells, and then we wash it because we want only the stuff that's sticking on the bottom to come up to tell us what the patient has. If there's a bunch of stuff floating around in there, it gives us mixed results. So we wash it every time. And then what we're going to do is we're going to purchase an immunoglobulin. So we're going to purchase an IgG, an immunoglobulin. So here is the foreign antigen. Here's the well. Let me do different colors. Here's the well. I have a cute picture of it, and I'll show you. Here's the well. So here is our patient's antigen that's going to stick to the bottom. So what we could do is, if we don't know what our patient has, we can fill thousands of wells, hypothetically, with the patient's antigen. So we can fill up tons of them at the same time. Super easy. So now what we're going to do is we're going to add an antibody that we bought from the store. So we're going to add this antibody, and this antibody, if it sticks here, then your patient is positive for this, whatever this antibody matches to that antigen. So if the antigen, if we're looking for influenza, we're looking for influenza. Does your patient have influenza or not? Well, we're going to design these antibodies to match influenza. If they stick here, your patient is positive. If they don't stick, your patient is negative. Does that make sense? So let's figure out how we read it. So what you're going to do is you're going to buy these antibodies from antibody.com. And on those antibodies is an enzyme called a chromatin. Is the enzyme called chromatin, or is the dye called chromatin? Yeah, chromatin. Oh yeah, the color changing, that's the substrate. Oh yeah. Okay, so it's just with an enzyme. So this is enzyme linked immuno. We have those three words already. Enzyme linked immuno. So here's the immunoglobulin linked to an enzyme. Then we're going to add a fourth thing, which is called the chromogen, apparently. And the chromogen is going to change color if that antibody. So now if you add the chromogen, it's going to change color. But let's look at what happened if your patient was negative. So here's the well. Here's some sort of antigen. Something's going to stick to the bottom. But it's not for the flu. This is not the flu that's stuck to the bottom because your patient doesn't have the flu. So what's going to happen is you're going to add these antibodies and they didn't stick because they don't match that. So the antibody didn't match the antigen. So regardless, so now what you're going to do, here's the enzyme linked to the immunoglobulin. When you wash it, all of these just come right up. So when you add the chromogen doesn't change color because it's not, there's no enzyme in there to change the color of the chromogen.
So it's reacting with the enzyme that's actually.
Yes, so the enzyme is what changes the chromogen's color. And if there's no enzyme, the chromogen won't change color. Are you guys with me on that? Let me explain it one more time. Stay with me. Your patient's antibodies, we added an immunoglobulin we bought from the store. This immunoglobulin matches whatever disease that we're looking for. And attached to these immunoglobulins is an enzyme. When we add the chromogen, because there's antibodies and because there's enzymes, that chromogen is going to change color, telling us our patient is positive. If our patient is negative, all of these antigens are here. We added the store-bought antibodies with the enzyme attached, but because they didn't stick to anything, because these don't match, because our patient is negative for the flu. So our patient's negative for the flu, these just floated around in there and did nothing. When we washed it, we washed those out. Now our chromogen is not going to change color because there's no enzyme in there to change the color. Are you guys with me so far? So this is called direct.
Is the enzyme reduced because of that?
The enzyme takes the substrate, so the Pac-Man takes the substrate, takes the chromogen, and changes the structure of the chromogen so that it'll change colors. Yeah. So like the Pac-Man eats the chromogen, and because the Pac-Man eats the chromogen, it changes the color of the chromogen. That's how the chromogen works.
Theoretically, you have a well with your patient's platform and put stored cotton
Is the screen moving for you?
different ones.
Yes. Okay. Very easy. We have the same neurological disorder.
Would you store by
It's getting busy.
antigen instead of the antibody?
No, like for antibodies, actually, so the antigen is actually patient in the body.
The antibody that's attaching to, we do one at a time. But they have machines that do it, and the machines will pick up 30 different types of antigens at a time, of antibodies at a time. Pick up those antibodies and drop them in all the wells. And is your patient positive for any of those? It's positive for influenza, not for Lyme, not for something else, not for something else. You know what I mean? Yes. Is that answer that? Okay, cool. Yeah, so this is a good way to test when you don't know what your patient has. You're just looking for all kinds of different things. Okay, so let's go back to an indirect. So an indirect, what we're doing is we're buying the microtiter plate. So we're buying all the wells that already have the antigen in them. So the antigen is already present. I really wish I could draw this somewhere else, but I'm just start right here. Hopefully you guys can see okay. So here, so a store-bought antigen. So, I'll just do one at a time. Okay, so here we bought this and we bought this from the same store. You already buy the well, it's already ready to go, it's already pre-primed with the antigen on it. So now what we're going to do is we're going to take our patient's serum, our patient's blood serum, and we're going to see if our patient's blood serum matches the antigen. So if our patient is positive, our patient's antibodies will match that exact antigen because our patient already has developed those B cell antibodies directed towards that antigen. Does that make sense so far?
So, direct is looking for antigen, indirect is looking for antibody.
Yes, yes. Indirect is looking for antibodies. Yes. So here, but how do we know? There's no enzyme. There's no chromatin. Yeah, go ahead.
So would indirect give you a positive result even like weeks after infection?
Yes.
And
Hypothetically,
direct would only
long.
if they had it at that time or in the
That's a good point. Yes. Yeah. So here's the antigen that we bought. Here's the antibodies from the patient. So don't forget those come from the patient. So now, but nothing's going to change color. How do we detect if it's going to change color? Well, now we need a second antibody. And this second antibody that this is the one we purchased from antibody.com. And these antibodies are attracted to human antibodies. So actually we isolate these antibodies from an animal. If you injected our blood into an animal blood, that animal would hypothetically die because you can't have foreign material in the animal's blood. So the animal will recognize that our antibodies are different than their antibodies. So that it would our antibodies are an antigen to their antibodies. So we're going to purchase these antibodies that are directed towards our antibodies regardless of what they match to. If there's antibodies there, I'll show you what happens if the patient's negative, but if there's antibodies there, the rabbit antibodies will match to ours. But we genetically engineered those rabbit antibodies to have a enzyme attached to them. So here's the enzyme linked immuno. Here's the sorbent part, they're absorbed. So here's our little enzymes. And then we're going to add the chromogen. So here, there's enzymes. So let's figure out what happens if our patient is negative. So here's our antibody, or here's our antigens that we already bought. Then what happens is our patient's serum comes in here. There are antibodies in our patient's serum, but they're not going to stick to anything. Because our patient doesn't have antibodies directed towards that antigen. Does that make sense? So, hypothetically, if we didn't wash that, we're going to wash it. Let's just wash it the way it works. So we wash it, we wash out everything else that doesn't stick, nothing's stuck. So now we're going to add the antibody. Yeah? Okay, so here, but they're not going to stick to anything. Because there's nothing for them to stick to. They don't recognize any antibodies. So we wash that out. We have the chromogen, it does nothing because there's no enzymes in there. Our patient's negative. Hypothetically, if we didn't wash it, if we didn't wash it, we'd have antibodies, human antibodies in here from our patient, but they didn't match to anything. If we didn't wash it, these antibodies from the rabbit would still match to the human antibodies. These enzymes would still be in here. And our chromogen would turn color, so it would look positive, but that's why we wash it, because wash it because there's nothing stuck to the bottom of the well.
Why would the antibodies stick to each other?
Why would the antibodies stick to what? To each other? Because this antibody recognizes this antibody. They stick that this antibody is directed towards that antibody. So just like an antigen, so just like if you have the flu, so here is the flu virus, and our antibody sees that as foreign. Here's our human antibody. The rabbit antibody sees that as foreign. Does that make sense? Okay. Yeah? Okay, so this one's negative. This is indirect. So indirect, we are looking for what? Antibodies. So what would we use this for? Why would we use this method? Well, this method is cool is if you are a clinical lab scientist and you are working in the HIV department. That's all you do. You look for HIV. Well, all the patient samples are going to come through, and you have thousands and thousands of wells that are directed toward HIV. You take a patient sample, you take 15 patients at a time, and you can test all 15 patients at once. Because all those 15 patients, if one of them comes up, you're like, that one's HIV positive. This one's HIV positive. This one's HIV. So you can look at a bunch of patients at once. This one you can look, if you're not 100% sure what your patient has, you can put the antigen in there, and then you can test for all things simultaneously. You can test for 5, 7, 12, hypothetically hundreds of different viruses at the same time. Yeah.
Sorry, I can't repeat it.
Oh no, please do. The more times you don't have it, the better.
Be testing the same virus for like doing analysis. And then indirect, you're actually testing for different viruses on the same thing.
So for direct, you can test for all kinds of different, you can figure out what your patient has. This one, you have to know. This is the HIV well. I bought this HIV well from the well store. And so I bought this HIV well from the well store. That's all I can test for. Because it's already stuck to the bottom. Does that help? Yeah? Okay. Okay, so let me show you my little pictures that I love so much. Oh, so here's the antibody. Remember, this is a great time to review what an antibody looks like. There's the heavy chain, the light chain, the variable region, the control region. We have 5 different control regions, or constant regions: IgA, IgG, IgM, IgA, GG, GM, D, E. Yes. Okay, so here's the antigen. Antibody binds. The enzyme changes the chromogen substrate. I love this little one because somebody made these little, like, I don't know, I really love it. So here's the antigen in the direct. So in the direct, this is from your patient. This blue is from your patient. This is from the store. This is from the store. Here's indirect. This is from your patient. This is from the store. So here's already, you bought this way. This patient's antibody. This is an antibody you bought from the store. Here's the chromosome. So what a titer is, titer means the concentration of a solution determined by titration. So that's kind of a funky way to say it. But basically, I don't want to define it exactly because it's not exactly, the definition is kind of used in a looser way. But titer, what it can mean is the lowest concentration that comes up positive. So how you can think about it, I think a good example is they did a titer on my daughter for chickenpox. Because she had the chickenpox when she was 3. And so they took her blood they centrifuged it, they took the serum, and what they did is they probably have a chicken pox, a varicella zoster, tons and tons and tons of them. I'm sure they do this all day, every day. So what they did is take her sample as well as 50 other patients, and they put her sample in here, and they looked at what concentration. You can also look at quantity. At what quantity did it turn positive? Does she have a few antibodies, or does she have a ton of antibodies? If she has a few antibodies, they recommend that she gets the vaccine and build her antibody storage. If she doesn't have very many, and it wasn't positive until a certain concentration, then that means that she needed the vaccine. If she was positive right away and had a lot of antibodies, then she didn't need the vaccine. So it's the minimum volume or concentration of antibodies needed to turn it a different color. So that's what titer means. Okay, so what is serotyping? So this one is important to know. It's important to remember this one because it'll come up again and again. But this is the same thing that you guys all did for your COVID tests, right? Did anybody not ever take a COVID test? Has anybody not ever taken one? You never taken one? So what you do is you'll put a swab up your nose and you mix it in saline solution, just mix it in some solution. And then you put some of that solution onto a little disc. And on that little disc, when you drop that little solution onto the disc, it's called litmus paper. And that litmus paper will pull the solution along in one direction. It's just like really absorbent paper that pulls moisture. So it's pulling moisture all the way as it goes along. So what is on that disc? What is on it? So on that little disc, over here is something called a control. And I don't know if you guys remember this, but if this one is positive, like this, regardless of what this one is, so this is where you drop it, and the litmus paper is going to go this direction. If this is positive, it means your test is not broken. It means your test works. What this is looking for are any antibodies at all. So this is almost like where they put the rabbit antibodies here. There's rabbit antibodies here. So if there are any antibodies whatsoever that cross that line, the chromogen is going to change color. The chromogen is already in the paper. That antibody is already in the paper. It's going to change color from it passing through that litmus paper. But here are the antibodies. Let me see if I can show you the results. Here are the antibodies that are directed towards COVID antibodies. So if your body has seen that strain of COVID, then those antibodies are right here. So here's antibodies that are directed toward antibodies for COVID. So those COVID antibodies have to match the antibodies. That will change color if it's positive. If it's not, if those antibodies don't match, it'll just keep going and you're negative. So it's the same idea. Yeah?
Is it pregnancy testing then?
Pregnancy test, you're testing for a hormone. So you're testing for, but it does work in the exact same way. So you're testing for the antibodies that will match the protein, or it's a protein, human chorionic gonadotropin, that will match and turn color. So yes, same exact thing. But you're looking for a protein instead of an antibody to coronavirus. Yeah.
So with the serotyping, it has the antigen on it. When
Yeah.
we're testing our antibodies, or it has antibiotic on there we're testing.
So they all work a little bit differently. So I don't want to make it one narrow way because it depends on which one you do. Like they have ones for all kinds of things. They have ones for Lyme disease. They have ones for
It was saying like IgM and IgG, how like how long you could duration.
Yeah, so they all work a little bit differently.
Where you're
but
So it could be either or.
it could be either or. Yeah.
Depending on like the specific color, then that would correspond to
It does, yeah. So it can be kind of a tighter situation. Like, is it really, really light, you know? And like, oh, well, I only have a few antibodies. It's really dark. I have a lot of antibodies. Yeah. Awesome. Okay, cool. So let's go ahead and get going. So let me show you what you need. So what we're doing today is a simulation. So it's not that much fun to do a simulation versus doing the real thing. But doing the real thing, we don't really mess with viruses, right? Viruses are like, yes. We don't really want to mess with patients' blood or serum or things like that. So I have seen labs that they do animal blood and that they'll test animal blood for antibodies and things like that. So it is kind of cool to do that. But we're just going to do a simulation. So what you're going to do is you're going to add. So you have 5 patients, right? Do you have 5 patients? Everybody has 5 patients? So you're going to make some assumptions about those patients in the questions. Oh, you have 6.
We have 6 now.
1, 2, 4, 5, 6. Oh yeah, you have 6. Okay, cool. So here you're going to add in your little plates. It'll have little pluses. So you add the plus fluid, plus fluid, plus fluid, minus fluid, minus fluid, minus fluid, A fluid, A fluid, A fluid, B fluid, B fluid, B fluid, blah 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 blah, all the way down. And those are simulated samples from each patient. So you have 6 patients, and you're testing those wells of 6 patients. So what we're mimicking is I think we're mimicking indirect. Or are we mimicking direct?
You already have the antibody.
No. But I don't know if they're supposed to or not. Maybe direct? Because don't we add an anti-antibody? Oh, we do add an anti-antibody. So I think we're mimicking indirect. So we're pretending that our wells already have the antibody on the bottom. Yeah.
You've got it all the way through F. Do we continue it all the way through H to the?
I think you only have A through F patients, right? Do you only have A through F patients?
Oh yeah, you're right.
Okay, cool. Okay, so those are your patients A through F. Your positive are positive controls to make sure that the titer is working, make sure the chromogen is working, make sure the antibody is working. The negative are negative controls to make sure that, I can't remember 100% what specifically in your negative control, but I'll figure it out. And then all your patients A through F. So on your little packet, it'll tell you how much to put into each well. Two things I wanted to mention. One is please don't put these anywhere except for in your beakers. So the lab tech said she don't want any of this stuff in there, so put these in your beakers. And your microtiter wells. So these will pop out. So you can pop these out when you're done. You can just dump them gently into your beaker. I think that's what she wants you to do. And then you just pop these off. And you can put this whole thing into the beaker.
It looks like
and
that's
then,
what people have done this morning.
Yeah, perfect. So you'll need pipette tips. You'll need a pipette. This is a great time to figure out your pipettes. What microliters are you looking at? Maybe converting that into milliliters and liters and going back from liters to milliliters to microliters, milliliters, liters. And just do some practice. I know there are probably 3 questions about that on the lab practical final. I will be happy to go over this with you. Everybody struggles with it, so it's a good time to just practice. So here are your positives, negatives, and then your patients A through F. Here is your primary antibody. So your primary, so you're doing antigen. Oh, because here you're doing your antigen. So we're adding our antigen. Then we're going to add our patient's serum. Do exactly like your book says, but it would make sense to add our patient's antibodies at that point. Hypothetically, we would wash it, but we're not doing that. Then we're going to add our secondary antibody. This is the rabbit antibody that matches the human antibody that has the enzyme already linked to it. This is the chromogen that will change color if your patient is positive, if it won't change color if your patient is negative. And I know there's questions about this on the lab practical final, so make sure you're really comfortable with it, you know what's going on, and you understand direct versus indirect, and I can go over it again, I'm happy to. So this over here is Lyme disease and influenza, over there is HIV and what do we have? Lyme and what? Oh, West Nile over here. So you can go ahead and